Hi, my name is Shawna Peebles, and this is an e-report on the review of Cell and Molecular Biology, Unit 3. This is a list of topics that will be discussed during this e-report. We will begin with the cytoskeleton and the protein filaments that are made up in the cytoskeleton. We will speak about cell crawling, specifically actin within cell crawling, and integrin, G-protein coupled receptors, whereas cyclic AMP and calcium are second messengers, cyclin-dependent kinases and cyclins and their role in the cell cycle, cell junction types and tissues, and cancer, sociopathic cells. The cytoskeleton is a network of protein filaments found inside the cytoplasm within a eukaryotic cell. The three types of protein filaments it is composed of are intermediate filaments, microtubules, and actin filaments. The composition of these filaments in the cytoskeleton aid in its ability for cell crawling, muscle contraction, and the difference in cell shapes during embryonic development. In addition, the cytoskeleton manages the location of the organelles and supplies a system for transport between them. Intermediate filaments are fibrous subunits found in both the cytoplasm and nucleus. Their specialized role is to provide tensile strength against mechanical stress. Mechanical stress can be pulling, stretching, squeezing, etc. Intermediate filaments resemble a rope-like structure. First, there are single fibers that intertwine to form strands. These strands then intertwine to form the rope-like structure. Within these fibers and strands are pairs of proteins bundled around each other to form dimers. When two dimers combine, a tetramer is made. These tetramers then wrap up together to give intermediate filaments their rope-like structure. There are four main groups of intermediate filaments. The first is keratin. It is the most widespread group of intermediate filaments in the cytoplasm. It is also the most diverse group of intermediate filaments because each keratin has a mixture of different subunits. The next group is vimentin and vimentin-related filaments, which are located in connective tissue cells, muscle cells, and support the cells of the nervous system. The third main group of intermediate filaments are neurofilaments, which contribute to most of the structural support of neurons. The fourth group of intermediate filaments are nuclear lamins, which strengthens the nuclear envelope of all animal cells. The nuclear envelope is held together by the interaction of intermediate filaments. Keratin filaments, vimentin, and neurofilaments are all cytoplasmic and rope-like. It is the lamins that are nuclear and form a 2D mesh. For this reason, lamins are very dynamic. They must disassemble and reassemble with each round of cell division. This is regulated by phosphorylation. Lamins are phosphorylated by kinases, which causes them to weaken their attraction to other lamins, therefore they disassemble. Lamins are dephosphorylated by a phosphatase, which makes them assemble. Accessory proteins, such as plectin, help to stabilize and reinforce intermediate filaments. These accessory proteins help to bundle intermediate filaments into a stronger formation. In addition, these proteins help intermediate filaments link with other components of the cytoskeleton to provide tensile strength against mechanical stress. This makes intermediate filaments more structural than functional. The second type of protein filaments I like to discuss are microtubules. The specialized role of microtubules is to organize the cytoplasm of eukaryotic cells. This is done by anchoring and localizing organelles, as well as providing a protein basis for cilia and flagella. Microtubules are more functional than structural. They're made up of long, stiff, hollow tubulin proteins. These tubulin proteins are dimers of two globular proteins. Each dimer has one alpha subunit, that is usually the minus side, and one B subunit, which is the plus side. Due to the interaction of non-covalent forces, the dimers stack together to form part of the microtubule. The entire microtubule structure is made up of 13 protofilaments with a chain of alpha and beta tubulin reciprocating in a straight line. These protofilaments have a beginning and an end. The beginning is alpha and the beginning is beta. Since all protofilaments face the same way, microtubules have polarity, where the ends are referred to as plus and minus. Tubulin dimers can be added to either of these ends. Therefore, microtubules form in vivo. The polarity of microtubules are highly important to their function. The two ends must differ both chemically and physically. Microtubules have a point of origin called a centrosome. The centrosome is a structure close to the center of the cell that microtubules grow out of. Centrosomes have a lot of ring-shaped networks that contain a third type of tubulin called gamma. This ring is the nucleation site or origin for mitosis formation. The negative end of the microtubule is enclosed in the centrosome, while the positive end of the microtubule is expanded toward the cytoplasm. Once a microtubule is nucleated, it begins growth from the positive end as new alpha-beta dimers are added to it. Suddenly, the microtubule falls apart by losing dimers off of the positive end. At the time, it only falls apart partially, but sometimes it completely falls apart all the way to the centrosome. This is called dynamic instability. Dynamic instability is driven by GTP hydrolysis. Each free tubulin dimer has one GTP molecule that binds to B tubulin. This hydrolyzes the GTP to GDP. The last protein filament I'd like to discuss is actin filaments. Actin filaments' specialized role is to help facilitate cellular movement. Actin filaments are thin, flexible, thread-like structures. They are made up of a twisted chain of globular actin subunits that are identical. They all have polarity, which points them in the same direction. Just like microtubules, actin filaments have a positive and negative end. In addition, actin filaments can grow on either the positive or the negative end. Yet growth is more favorable on the positive end. Actin filaments need to bind to associated proteins in order to maintain their stability. Without this association, they would disassemble. First, a free actin monomer binds to ATP, and once it is integrated into a filament, the ATP is split to ADP. This causes a reduction in the strength of binding the monomer with the filament, stimulating depolymerization. For this reason, actin filaments only last for a few minutes unless they bind to an associated protein. There are small proteins in a cytosol that bind to actin monomers to keep them from linking to growing actin filaments. Actin filament polymerization is controlled by these small proteins as they keep the actin monomers free. Once actin filaments are made, they are held together in parallel bundles by actin bundling proteins. Actin filaments are then combined into a protein network by actin binding proteins. There is an actin-rich cortex underneath the eukaryotic cell membrane. This helps the membrane have mechanical strength and determines the shape of the cell. It is the reorganization of actin in the cell cortex that determines a cell's ability to move, change shape, etc. Moving on to topic 2, I'd like to speak about cell crawling. Actin is necessary for cell movement. Cell crawling involves a 3-step process, and each process uses actin differently. The first step is when the cell has a protrusion in its front end. This is guided by new actin polymerization. Lamellipodia are thin structures, yet they contain a lot of actin, which is close to the plasma membrane. Filopodia are even thinner structures containing actin. Both lamellipodia and filopodia are involved in the protrusions of actin during cell crawling. The second step is for lamellipodia and filopodia to adhere to the surface in which the cell will crawl. This step is completed by the linkage of a transmembrane protein called integrin and the actin filaments of the cell's cortex. This adherence helps to anchor the cell to the surface. Step 3 is the contraction of the rest of the cell, in which causes the movement. Contractions occur when motor proteins called myosin bind to actin. When myosin moves across the actin filaments, they travel from the negative end to the positive. This is beneficial to cell movement because the protrusions discussed in step 1 occur at the positive end. So when myosin moves from the negative end to the positive end, it furthers these protrusions, causing the cellular movement. During this signal transduction, a signaling molecule binds to a 7-transmembrane protein. At the end of the 7-transmembrane protein are 3 subunits, a gamma, beta, and alpha subunits. Alpha lets go of GDP and binds to GTP. The alpha subunit containing GTP moves along the cell's surface. Then they both bind to phospholipase C. This activates or turns on phospholipase C, which then activates PIT2. PIT2 is then hydrolyzed or split into IP3 and DAG. DAG moves along the cell surface, which activates protein kinase C, which causes calcium channels on the outer surface of the cell to open and calcium rushes into the cell. But IP3 enters the smooth ER and activates calcium channels within the cell, which open up and even more calcium rushes into the cell. This increase in calcium levels within the cell activate calmodulin. Calmodulin then activates a CAM kinase, which then causes cellular effects. Moving on to the 5th topic, I would like to discuss CDKs and cyclins as part of progressing through the cell cycle. First, let's begin with a very brief review of the cell cycle. The cell cycle has 4 phases. G1, or the GAP1 phase, S, or synthesis, G2, or GAP2 phase, and in the M phase, or mitosis. In the G1 phase, cells do most of their growing. During S phase, chromosomes are replicated and DNA synthesis occurs. During the G2 phase, many of the organelles and molecules are made for cell division. All of these three phases make up interphase. The M phase is where mitosis and cytokinesis occurs. And there are 5 stages within mitosis: prophase, prometaphase, metaphase, anaphase, and telophase. The cell cycle has 3 main checkpoints. During the G1 checkpoint, there is a check to see if the cell's environment is good for division. During the G2 checkpoint, there is a check for proper DNA replication and repair. During the N checkpoint, and specifically in anaphase of mitosis, there's a check to make sure that mitotic spindles are attached to sister chromatids. However, the overall control of the cell cycle is dependent on cyclically activated kinases through phosphorylation and dephosphorylation. So, cyclic regulation is carried out by kinases and phosphatases because they are always being expressed. Kinases and phosphatases promote the cell's growth throughout the cell cycle. So, here's a graph depicting how cyclins control kinases. Right here are the kinases. These dotted structure are the checkpoints. Here's the different phases of the cell cycle. And this structure right here is representing the cyclins of each cell cycle phase. Now, although kinases control the proteins of the cell cycle, there are proteins that control kinases named cyclins. These cyclins bind to kinases, then activate them. These kinases then become cyclin-dependent kinases, or CDKs. The difference between kinases and CDKs is that kinases are constitutive or constantly expressed, as the graph here depicts, whereas CDKs have to wait on a specific cyclin to be made before it is activated. And in order for a cell to move on to the next stage in the cell cycle, a cyclin CDK network has to promote cellular activity. And it is the phosphorylation of a kinase that causes the cellular reaction. There are proteins that inhibit CDKs to stop the cell cycle at checkpoints. These proteins are called CDK inhibitor proteins, which prevent the interaction of CDKs and cyclins. This stops CDKs from being activated. The cell will then pause at a checkpoint. All in all, if a cell's environmental cues favor cell growth, then the inhibitor protein is removed from the CDK. Yet, if the cell receives an environmental cue that is unfavorable for cell growth, it is defaulted into a stage of the cell cycle called G0. Moving on to topic 5, I will speak about cell junctions and their types and tissues. Tissues are made up of cells and an extracellular matrix. Integrin is a transmembrane protein that connects extracellular matrix proteins to the intracellular cytoskeleton of cells. Cell junctions link together cells in the epithelial sheets that outline the surfaces of an animal body. There are 5 types of junctions. These are their proteins and functions. The first type of junction I'd like to discuss is the tight junction, which is formed by claudins and occludins. Their main function is to seal cells that are close to one another inside of an epithelial sheet. This serves to stop leakage of the extracellular molecules between them. In addition, it helps to polarize cells and may form like a zipper. Next is the adherens junction. Adherens desmosome and desmosomes both utilize cadherin proteins, yet cadherins bind to actin filaments and adherens junctions. The main function of an adherens junction is to join actin bundles in one cell to that of another. So, as stated before, desmal junctions also use cadherins, and the cadherins within desmosomes bind to keratin filaments found in the intermediate filament family. This main function of a desmosome is to join intermediate filaments in one cell to that of another. Gap junctions link together transmembrane proteins called connexins to form a channel between cells. This functions to allow small intercellular molecules to pass from one cell to another. And the fifth type of cell junction is hemidesmosomes, which use transmembrane protein called engrin to link collagen. Its function is to join an epithelial cell to the basal lamina.